students, welcome back. Today I am Anuradha from DPS Haldwani Amateur going to start another topic of the same chapter, that is human health and disease. So students, in this chapter we have covered about the definition of disease, okay, and the classification. Then we have covered that how our body is attacking for these pathogens. So what is required? Immunity is required. So immunity can be of two types. What are they? Innate immunity as well as the acquired immunity. And innate immunity, what we have done, the immunity which we are getting from our birth, okay, that one is the innate immunity. And the second one, that is the acquired immunity, that type of immunity which we are getting from our environment after our birth. Okay, so here if I will talk about this acquired immunity, what it is showing? It is showing that if any pathogen, okay, if any pathogen, it may be bacterial or fungal, any disease pathogen will attack the cell of any individual, what they will do, these cells or the things which is present here, they will make some proteins. We have done this one. So this is preparing the antibodies for these antigens. Antigens are what? Those pathogens which are attacking the individuals, they will become the antigens, and in response of this antigen, the body will form what? They will form the antibodies. So what you will call it? It is showing the response. Okay, so this response can be of two types. What are they? This can be the primary response and this can be the secondary response. So now question arises that what is this primary response and what is this secondary response? Now if we'll talk about the primary response, example is that the first time if you are getting any pathogen, or we can say the first time any pathogen encounters or any body encounters with this pathogen, so that first time the body will show the response for the particular pathogen and they will form the antibodies. So this type of response, it is known as the primary response, right? Now the secondary response. Secondary response means that when any of the individual or the body encounters with this particular pathogen second time or third time or later on, okay, or we can say subsequently, when it will encounter with the particular pathogen and on the response of this pathogen they are forming the antibody. So what it is? Secondary response. Okay, so examples we have done that, for example, chicken pox. Okay, the first time chicken pox with an individual is having, what at that time the body is preparing antibodies for that one, and after that one again it will try to attack the body. So what body will do? Body will make more and more antibodies and your body will not be affected by this pathogen again. Okay, so we can say that this primary response is what? It is somehow slow, because the first time when it is encountering the body, what happens? The body will take time to respond for that particular pathogen. But when we will talk about the secondary response, they are somehow fast. Why? Because now body know about how they have to respond for that particular pathogen, right? So whether it is a primary response or the secondary response, both are controlled by the lymphocytes. How? Because we know that this lymphocytes can be of two types, B lymphocytes and T lymphocytes. In the previous lecture, we have done about this B lymphocytes and the T lymphocytes. Here, if we'll talk about this B lymphocytes, the antibodies will form in this B lymphocytes or the B cells, and for their maturation, okay, for their maturation, they are going to T lymphocytes, or we can say to the thymus. Okay, thymus is what? Thymus is a gland, okay, which is present just beneath the, we can say, our heart. So these B cells, after forming the antibodies, what they will do? They will mature here, but more maturation will take place in this T lymphocytes, right? So like this, by making antibodies or with the help of these cells, they will attack or they will protect our body with the pathogen. Okay, so here we have done about the basic thing. Now comes to the antibodies. As we are saying, from these B lymphocytes, okay, because B lymphocytes are what? They are preparing the antibodies. What they are doing? They are preparing the antibodies, and these antibodies is forming where? Where in the bone marrow. Okay, if you will talk about the long bones in our body like the femur, so what happens? There will be formation of these types of cells, and in these cells what happens? There will be formation of more and more antibodies. So now when we are talking about these antibodies, what should be the structure? Okay, so what is the structure of antibodies? Here I have drawn this diagram of antibodies. Okay, how you can say that it is an antibody and how it is forming? See that every antigen, okay, every antigen having some different types of structure. Okay, so that's why this antibody structure can be somehow changes where into their this site. Why? Here you can see that structure of antibodies Y-shaped structure. If I will ask you what is the shape of the antibody, it is a Y-shaped structure. Now this Y-shaped structure contains what? They contain two small and two longer one. Okay, what you are seeing? Two smaller and two longer one. What it is? They are these smaller one. It is known as the light chain. Okay, so this one, the second one which you are watching here, what it is? It is the light chain, and this complete one. Okay, this one, this is known as the heavy chain. Okay, so this is the heavy chain, two heavy chain and two light chain. That's why we are saying H2L2. Okay, two heavy and two light, and this is the representation for the antibodies. Now, as you know that antibodies is what? Antibodies is immunoglobulin. When we are talking about this antibodies, they are immunoglobulin. It means that they are having some globulin structure. It means that they are having some proteinaceous structure. Okay, so now these structure having two disulfide bridges. Okay, here you can see that these sulfide bonds are present. So what they are showing? Each is having what? They are having the disulfide bridges. So I can write here disulfide bridge. Okay, two disulfide bridge. And if you will count these one, what you will see here? 1, 2, 3, 4, 5, 6, 7, 8, right? Then 9, 10, 11, 12, 13, 14, 15, 16. Okay, so how many they are having? They are having the 16, right? So now we have done that what is the structure of this antibody and how they are connected. One more important thing, we are saying that these antibodies, what is the role of these antibodies? If any of the pathogen, for example, this is a pathogen, if any of the pathogen comes towards this and towards our body, okay, when this pathogen comes to our body, so these antibodies, these are the antibodies which are released from our body. What they will do? They will bind with this antigen. Okay, what they will do? They will bind with this antigen. If I will show this diagram here, so these are the antigen. Okay, so what they will do? They will bind this antigen inside it, right? So when there will be this binding, so this site, what you will call? You will call the antigen binding site. Okay, in the antibodies, that site where antigen is binding, it is known as antigen binding site, and this is present here also as well as this site also. So this is also a antigen binding site, right? And these antigen binding site, they are known as what? They are known as the paratope. What you will call? Paratope. And that part of antigen which is binding with this antibody, it is known as what? It is known as paratope. Sorry, epitope, right? So this is epitope and that one is the paratope. Okay, and one more important thing, that the lower part, This lower part will be remain stable of the antibodies, but they can be changed accordingly, right? So they are not the stable one, they can be vary, and these are the stable one. Okay. Now, as I have said that they are showing the response, and these responses again we can classify into two types. What are they? Humoral immune response as well as the cell mediated immune response. Okay. In our body, if any pathogen is coming towards our body, so some cells are there which are attacking these pathogen, and some antibodies are attacking to the pathogen, right? So on the basis of these responses, we have categorized into two. That the first one is the humoral. Humoral means something which is related to what? Fluid. Okay, something which is related to the fluid, it is known as the humoral. So the response, okay, that response or the immunity which is provided, it is due to that fluid. Okay, and what is that fluid in our body? It is a blood. Okay, so we can say that the blood or somehow lymph, they are responsible for what? They are responsible for providing the immunity to the body for the pathogen. It is known as humoral immune response, right? And it is also known as the antibody mediated. Why we are saying antibodies? Because these antibodies are prepared by what? By the preparation of this lymphocytes, right here. So we can say that these lymphocytes are what? The lymphocytes which is present in the lymph. Okay, and these are the antibodies, and we are having 5 types of antibodies. What are they? This IgD, then GAME. Okay, all these are the immunoglobulins. Okay, so when we are talking about all these antibodies, these are the immunoglobulins. So we are talking about the humoral immune response. Okay. Now, one more important thing about it, that it is related to what? It is related to the, we can say that the B lymphocytes as well as the T lymphocytes, as we have covered here. Okay. Now the another one is the cell mediated. Here what is responsible? Fluid is responsible. Here cell is responsible. Okay, in our body some cells are also there. They are providing the immunity to our body against the pathogen. So what are they? Okay, so here we can say that about the T lymphocytes. If I will talk about the T lymphocytes, what are they? They are having this cell mediated response. Okay, CMI, cell mediated immunity response. So how they are acting? How they are acting in our body? In our body, okay, in our body we are having these B cells and the T cells. The role of B cells forming the antibodies, okay, and the T cells, they are having again something related to the cell, and they will function as a we can say cytokine, okay, as well as they will act as a helper, or they will act as a suppressor or the memory. Okay, so role of this cell mediated is what? It can be act as a killer cell, it can be act as a helper cell. I am denoting here like this. Okay, TC, cytokine, it is known as the killer cell. It is known as the killer cell. And this one TH, it is known as the helper cell. It is known as the helper cell. And third one TS, it is known as the suppressor cell. It is known as the suppressor cell. And the fourth one TM, it is known as the memory. Okay, so on the basis of their names, they are helping our body for the particular pathogen. For example, if any pathogen will attack our body, okay, if any pathogen will attack our body, so antibodies are there, okay, antibodies are there, but some cells are also there which will attack on this pathogen. How? In the T lymphocytes, okay, when we are talking about the T cell, which is present in the thymus one, what they are having? First of all, they will secrete 3 types of T cells. Okay, they are having 3 types of T cells. The role of this cytokine, what it is providing? It is providing some this type of CMI, cell mediated, or we can say graft rejection. For example, if I say I have to transplant, if I have to transplant any organ, example kidney, from one person to another person, the tendency of our body, we can say that it is not taking any other cell. If any cell or any of the things which will come from outside in our body, so what is the role of this one? They will detect that these are not the self one. Okay, they will not harm to the self cell, but as any other cell which is coming from our outside, they will act upon it. Okay, so that's why when we are going for the transplanting of any organ, what we are doing? We are going for the histopathological test. Why? It shows that, or it should give the idea that whether we can transplant this organ or not. Okay, and along with this, some blood tests are also there. Why it is required? That if any organ we are transplanting just like this, so body will not accept that tissue. As a result, it will again fail. So before transplanting, what we are providing? We are providing the immunosuppressants. What we are providing? Immunosuppressants. It means that thing, that immunity, that will suppress the effect of the self cell. Okay, so what we are giving to those patients? First of all, we are starting or we are giving those patients the immunosuppressant, so that their body cell will not be do that much activity, so that it will not do that much activity to detect any foreign cell. And that time we are transplanting, and even after the transplanting, we are going to give this immunosuppressant, so that it will not reject the outer cell, right? So that's why we are saying that this is a graft rejection. If any of the graft or any of the tissue we are providing from outside, it will reject it, because it is having the tendency of the self recognition. Okay, it will recognize that whether it is a self cell or any foreign cell. So these TC, what they will do? For example, here we are talking about these organs, which is required one. But sometimes what happens, if any of the pathogen will attack in our body or in a healthy body, so these cells, what are these? These cytokine cells, what they will do? They will start producing some of the things, so that it will kill that cell which is coming from the outside. Okay, it will kill the cell of the pathogen, and pathogen will not able to grow inside the body. So the first one, it will kill that one. Another one is the helper cell. So helper cell, what is the role of this helper cell? Helper means that it will take help from any other cell, and after this they will respond for that pathogen. For example, again, if any pathogen attacks to the body, what it will do? It will give signal to this killer cell. Okay, it will, for example, it will give signal to the killer cell that come here and just kill that pathogen. As well as it will give invitation, or it will say to these B cells, okay, which is present here, which are making the antibodies. What is the role of this one? Antibodies. So this helper cell will send the invitation to the killer cell as well as the B cells to come here and to attack this pathogen and kill it, right? So here we have done the work of helper cell. Now the third one is the suppressor cell. Now sometimes it happens that during the fight of these killer cells, antibodies, while they were fighting with that pathogen, they forget to stop that reaction. Okay, now sometimes at some point it is required that now the defense mechanism should be stopped because the pathogen has been killed. So that time the suppressor cell act upon these cells, and it will suppress the reaction of that antigen and antibodies as well as this killer cell, right? Now the role of memory cell. What is the role of memory cell? As I have said, the first time if any of the pathogen entering inside the body, so now these memory cell will having particular memory for that particular pathogen. If in the subsequent time they will attack to any body, so these memory cell will become activate and they will recognize that this is that pathogen, and we have to make such type of antibodies or the cell against that one pathogen, right? So by this type, the antibodies which is forming by the B cells, okay, we have done about these B cells, they are forming the antibodies, different types of antibodies they are preparing for the different one, and with the help of these cells we are providing some immunity to the individual. Now total here we have done two types, humoral which is in the blood, and this cell mediated which is provided with the help of cell here. Now we are having classification for the immunity, whatever the immune response they have responded. So on the basis of this we are having two types. What are they? Active immunity as well as the passive immunity. So now what is the meaning of active immunity? See students, that any immunity or any defense mechanism which is happening by the body itself. Okay, what I am saying, the immunity which is provided by the body itself, it is known as active. Okay, passive immunity is that response or that immunity provided by the others in your body. That one is the passive immunity, right? Examples you can say, if you talk about this active immunity. Active immunity, as I said, your body is making. So example, chicken pox. Okay, so you are not going to give any of the vaccine for that. What you are doing, just the first time if any of the pathogen attacks your body, so from the next time it will not be able to attack again. Why? Because your body will make the antibodies for this chicken pox, right? So like this comes under the active immunity, because your body is making antibodies for that particular pathogen, right? And this one is the example of natural active immunity. It can be sometimes artificial active immunity. How? When we are providing some vaccination. Okay, so first time we are providing vaccine to the body, so that this, if anytime this pathogen will come in contact with the body, so your body will start making antibodies for that particular pathogen. But again, what is happening, your body is making the antibodies, no one is going to make the antibodies, only we are giving the facilitator as a vaccine, but the preparation done by the body itself, right? Now what about the passive immunity? Passive immunity means that someone is making antibodies and we are incorporating direct inside the body and they are responding. For example, if we'll talk about the natural passive immunity, okay, we know that we are having different types of antibodies which we have discussed here. Among them, if you talk about this IgA is what? It is also an immunoglobulin or the antibodies which is present where? In the mother's milk, or that milk which is the first yellowish milk which is known as the colostrum. That's why it is said that we have to give the baby this colostrum, or this the first feed or the initial lactation which contains the more amount of this antibodies. Okay, so the babies, they are getting immunity from where? From the mother's feed. The baby is not able to make antibodies by itself, so what it is doing, it is taking from the mother's milk. So the antibodies prepared by the mothers, it is taken up by the babies, so it is example of passive immunity. Same thing, that IgG, this one is very very important. This is the only antibody which will pass through the placenta. Means that this antibody, when any of the fetus or the babies are inside the mother's womb, so how they are taking, or how they are their body is fighting with the pathogen with the help of antibodies. So this antibody will give protection to the babies in the mother's womb, and this is the only antibody which can pass through the placenta and providing protection to the growing baby. right? So these two antibodies, this is example of natural passive immunity. And what can be the example of another one of the artificial passive immunity? If we will talk about the tetanus, okay, or if we will talk about the snake bites, right? So at that time our body needs antibodies, but if it will make naturally, it will take time to make the antibodies. So at that time what we are providing, we are providing them the injection which is having antibodies for the tetanus, okay. So directly we are providing that antibodies inside the individual. Example, in case of snake bites, directly we are providing the antivenom so that it should be work very fast, right? So here also we are providing immunity from the outside, right? So like this, what we are doing, we are providing the immunity from the natural one as well as the artificial one. So now, as we have said that there will be two types of immunity, active immunity and the passive immunity. So the active immunity is done by the body itself, but the passive immunity, in which we are giving the antibodies from outside, what are they? This will be the part of the passive immunity, okay. So this is what is providing from the outside, what it is in the form of vaccination, okay. In active also, what we are providing in the artificial form, the antibodies which is prepared by in outside, and again they are provided to the body here. So what we can say, what is this vaccination? Vaccination is the method, okay, and in this method what we are injecting, we are injecting the vaccine. So what is this vaccine? The vaccines are the thing, they are the we can say antibodies, okay, which the antigen will form. But what we are doing, we are preparing those antigen, okay, we are preparing those antigenic proteins which is taken from the pathogens, okay, which is taken from the pathogen. And this pathogen can be live or the dead or the weakened one, okay. So that's why I've written here that the proteins or the antigenic protein, okay, because for our the body, for any individual, these protein are what? These are the foreign cell, foreign material. So what we'll call it, antigenic protein. So this antigenic protein, they are taking from the pathogens, okay, and these pathogens can be live one as well as they can be the inactivated form or they in the weakened form, whatever, right? So from there we are taking those proteins, and after that we are injecting those protein in the injection, and we are injecting this one to the individual. Why? Because as first time when we are providing these antigenic protein, what we are providing, we are helping our body to form the antibodies against this one, against this pathogen. So now our body will be able to recognize that structure of, or we can say constitution of this pathogen, and when in the subsequent part, or when anytime this pathogen will try to attack the body, so at that time body can recognize very easily and they will act upon those pathogen and they will make more and more antibodies for that particular pathogen, right here. So what is it? This vaccination is what? When we are providing this vaccine, and vaccine is what? When we are forming this vaccine with the help of the extracted antigen from those pathogen and injecting to the host, or we can say into the individual, not the host here, right? So what is the role of these antibodies? So these antibodies produce antigen, okay. So what they will do, antibodies they are having, and they will neutralize the effect of that particular pathogen which is going to attack the body, right here. And we know that they are having memory cells also. We have done this one, TM, okay. So this antibodies or the cells which is present in our body, what they are having the memory cell, and these vaccination, what it is doing, it is generating the memory for any of the antigen with the help of B cells and the T cells, as we have discussed right now. So now example I have mentioned here, tetanus. Okay, so that in the tetanus, what we are providing, we are injecting any of the body with the what, with the this antigenic proteins, right? So what it will do, and here what type of microbes we are taking, we are taking dead microbes. Make sure that you must know that we can take live, dead, or the weakened one. So for making the vaccination for this tetanus, okay, what we are doing, we are providing the dead microscopes injected into the body, so that the body will make the antibodies for this pathogen. Right here, same like this snake bite, as we have discussed that as um, snake bites to any individual, what is required, we cannot wait for our natural body to make the antibodies. Why? Because at the time we need to take a quick decision, okay, very fast one. So what we are providing them, we are providing the antitoxin, okay, which will act against that toxin which is by the snake. So very fast it will attack on that pathogen, and or we can say on the toxin, and what it is providing, it is providing the passive immunization. Are you getting? Passive means that you are providing to the body, right? So what, how can we say that this vaccination or immunization? Okay, vaccination, where we are providing any of the pathogen so that it will memory for that pathogen, and in the immunization, just right as the pathogen will attack, okay, or any toxins are there, then we are providing against that particular pathogen or for the against that toxin, right? So it is a part of immunization. Now some more methods are there in today's world. For example, recombinant DNA technology. In this recombinant DNA technology, we are removing the DNA from any of the pathogen and injecting any of the healthy individual with that DNA, so that if another time it will act upon the body, so our body will able to understand the mechanism, and it will make more and more antibodies, and it will help our body from that pathogen, right? And this recombinant DNA technology is done for the hepatitis B, okay, and which they extracted from the yeast cell, right? So this is for your MCQ, you have to know about it. Now as we are talking about this immunity, about this vaccination, immunization, we know that some more affect our body, okay. See, as pathogen will attack in our body, so body is showing response in the form of making antibodies, they are helping our body. Sometimes what happens, sometimes some more antigens are attacking our body, okay. So this attack which is happening here, so our immune system will not be able to cope up with that pathogen. So at that time what it will show, it will show the allergy. So allergy is what, the exaggeration of our body, of our immune system, okay. So we know that our immune system is what, they will detect that this is the foreign pathogen, or it is having the capacity to recognize the foreign cell. So here our immune system is so much responding, even if any of the antigen of any of the things will come in contact with our body, so our body is showing the response for that one, and this response is in which form, in form of allergy, right? So the same thing written here, the response of immune system to certain antigens, okay. So certain antigen which is present in our environment, as they will come in contact with our body, so our body will show some response in form of this allergy. Now another term is the allergens. Before that we have done mutagens, okay, those agents which are responsible for mutation, those are mutagens. Same thing here, allergens. Allergens mean, aller means there's something related to the allergy, and they are generating it, right? So the substances which are responsible for the responsible of the immune response, okay, because our body is responding for that particular antigen. So those agents which are activating it, what you will call it, you will call the allergens. Examples are pollen, dust, Okay, so these are some of the things which is present in the environment, and you must have aware about this pollen. Okay, sometimes the people are very sensitive for these dust and these pollens, and along with this some clothes also. So what it is affecting, it is affecting our immune response, right? And those antibodies which are responsible for this allergy, it is the IgE. Okay, we have done different types of antibodies here. So this Ig is what it, when there will be any of this allergens, so our body will make more and more antibodies. So what type of antibodies it will make? It will make the IgE. Okay, now what can be the symptoms? Symptoms, you know that you will feel like sneezing, watery eyes, and the skin will become reddish, inflammation may be there, or running nose, or there may be some difficulty in breathing. So these can be the symptoms which can be due to the allergy. Okay, and why it is happening? Why it is happening? If I will talk about these allergens, so if allergens will come in contact with any of the cell, okay, if allergen will come in contact with any of the cells, so the cell which is present in our body, okay, so that cell is the mast cell. Okay, what is it? The mast cell which is present in our body, what they will do? So for example, this is a pathogen. This pathogen attacks and it will go to the mast cell. What it will do? The antibodies which has been prepared our body. What type of antibodies? Our body will form the IgE. Okay, so these antibodies will attach with that pathogen. So this will attach with this pathogen here. Now these will enter where? In the mast cell. So as a result, what will happen? This mast cell will release some of the substances, okay, or we can say degranulation. So the granules which is present in the mast cell, they will come outside, and in which form they will come here? In form of this histamine and serotonin. Okay, are you getting what it is secreting? In form of histamine and serotonin. So now what is the role of this heparin and serotonin? See student, one of them is the vasoconstrictor and one is a vasodilator. Okay, vasoconstrictor means the vessels will become increased in size, or we can say the diameter of these vessels will increase, and vasodilator means they will reduce. So in both the condition, whether the vessels will be dilate or the constrict, what will happen? There will be some movement of blood which is present, which is happening inside these vessels. It will be very difficult to pass on. Okay, due to that, what will happen? The body will show some allergy right here. So what are they? This histamine and serotonin which has been secreted by the mast cell. Right, same thing written here. Due to this allergy, they will release the histamine and the serotonin from the mast cell. Okay, and this mast cell attacked by the pathogen, as well as this antibodies will attach with this pathogen to show the response. Right here. So what can be the drugs which we can use here? Antihistamine. Antihistamine means which will attack against this histamine. Okay, so antihistamine, adrenaline, and some steroids we can use for the allergies. Right here. So today we have completed that much. So that's all for today. Thank you.